We're going to be talking about some pilot testing that King County participated in for source tracking. We did this work this summer. Jeannie's going to talk about a mobile water kit. And then Andrew and I are going to be sharing the stage talking about the canine scent tracking team that we hired and human Bacteroides qPCR method. So Jeannie's going to start.
My name is Jeanne Dorn, and I am one of Debra's colleagues at King County. I'm in a different part of King County than Debra, though. She's in the science group, and I'm in the stormwater services group. So I'm so happy to be here to tell you about a pilot test that Stormwater Services conducted in June of 2014 of a rapid bacterial assessment technique. And this rapid technique is called a mobile water kit. It was developed by researchers at the University of Alberta in Canada, and they developed it because there's a real pressing need to assess the safety of drinking water wells in remote parts of India where they don't have the money or the labs for lab testing. Very high infant mortality from drinking bad water. So that's why they developed it. We were excited to learn about it because we could use it for our surface water and stormwater bacteria screening and source tracing programs if it works. So we were fortunate to actually run a pilot study of it in June of this year, as I said. And what we did was we set up the test to obtain about 50 samples, real-life samples from 4 different parts of King County, an urban center, an urbanized center called White Center, a very rural agricultural basin called Boise Creek, and then streams and lakes from North King County, and I think also conveyance systems, no, I can't remember, streams and lakes from North King County, and then City of Kirkland also participated, and they had both stormwater pipe system and streams. So the little picture on the right lower shows what the test actually is. It's a very inexpensive test, we hope, when it's actually commercially available. It's a little small filter through which 100 mils of water is pressed through, your sample water, and then very small drops of 4 different reagents are then put on the filter. And if there's bacteria present, there'll be a red color. If there's a lot of bacteria present, there'll be a lot of red. So it's semi-quantitative. That's kind of the goal of it. I'm going to skip these two. What were our results of this test? Well, over 2 days, we obtained 50 samples. We assessed it with the mobile water kit in the field, but we also sent the samples to our lab for analysis for E. coli. So what are our findings? Our findings are that there's a lot of promise for this kit. They're on the right track. In some of the cases, sorry I don't have a pointer, but the green line in this slide would be perfect agreement. If the test analysis agreed between the mobile water kit field test and the E. coli, which is on the y-axis, you'd want to see all your samples clustered on or very close to the green line. Well, we didn't quite get that. It's not quite there yet. You can see quite a few samples are over on the y-axis, and they had no, they were indicated to have no bacteria by the mobile water kit. So it's not quite there yet for a number of technical reasons, which I don't have time to go into. However, they actually had a lot of turbidity in them, and that was one of the cool things we encountered in the field. When the test kit was made, it was for drinking water, which is clear. So just doing this test in the field was very valuable for the researchers because they saw, oh, turbid water doesn't work quite so well on. So there were some technical difficulties, but we're very excited to have done this test. It's a cool pilot test that we ran, and hopefully the university researchers can work to refine their test with our input, and we'd be very happy to run another pilot test if they further refine it. And I'll just show you, this is actually what the mobile water kit looks like. You can do 3 samples at a time. You can force 100 mils of each of 3 samples through the mobile water kit and then drop your reagents. And within about minutes to 45 minutes, you have your result. So it's very cool. It's got promise, and very important, these people made this test possible, and there's people in this room who worked very hard on it. So thank you very much, and I'll turn it back over to Deb.
Andrew and I are now going to talk about the canine scent tracking that we used and the human Bacteroides. We participated in this with Seattle Public Utilities. City of Kirkland, and Environmental Canine Services was the team that we hired from California. And fortunately, Ryan and Jonathan are here in the audience, so if there's any questions in particular about their watersheds, they can help answer those. So just to give a little background, we started off with, all our monitoring starts with fecal coliform or E. coli, and if we get a high value, it leads us to the usual suspects. And that could be problematic if we're trying to manage for a resource. For example, we sample swimming beaches and we have high bacteria. We don't know, is it waterfowl? Is it a dirty diaper in a toddler? Is it the tributary leading into the beach? If we go up the tributary and we have high bacteria, we don't know, is it a side sewer? Is it an illicit connection? Is it dog poo? Is it raccoons? And if you get in the rural areas, you have the same issue with cows and agriculture and deer and elk. So the King County Environmental Lab has been working on fine-tuning the qPCR technique, and I'll talk about that a little bit later. And they now have a test for ruminants and humans. And we chose this marker. It's the second generation of the qPCR testing that our lab's been doing. And then we had heard about the canine scent tracking team in California, so we hired them to come and do a comparative study. We chose these two watersheds. They both drain to popular swimming beaches in Lake Washington, and they have a history of high bacteria counts. They have a high counts in both the creek and at the swimming beach. Both creeks have had intensive bacteria investigations funded through the Department of Ecology through grants, and we have a history of working in partnership with the City of Kirkland and SPU on these investigations. There's no TMDL on these creeks. We're doing the direct to implementation strategy that ecology has approved. Well, it's not an approved plan, but you know what I'm talking about. So our study design was restricted by the fact that the dogs were coming from California. They can only work 4 days in a row. We didn't know how long their sniffers would hold out. It depends on the intensity of the smells they're smelling. So we divided the 4 days into two watersheds, doing Juanita Creek first and Thornton Creek the second day. The canine handlers suggested having field crew go out into the watershed and collect samples first and meet everybody at a scent neutral field where we poured the samples into these plastic tubs, and the dogs could run through and smell the buckets, and then from there we could go to site investigations later on. And here you can see a picture of taking the dog into the watershed and doing some catch basin, flipping that. Every place we sampled, we collected a separate bottle for laboratory testing, and you can see a picture there of the field crew person collecting 2 bottles. And even when we were out in the field doing site investigations, we collected a bottle to go to the lab, which actually slowed the dogs way down. They could have moved a lot faster and covered a lot more territory if we weren't collecting water, but we wanted to do this paired sampling strategy. We had 4 controls and 4 sets of controls. Our positive controls consisted of diluted influent from the West Point treatment plant. We had 4 negative controls, which were distilled water. And then we had 4 alternate waste samples contributed by volunteers. And then we had 2 field duplicates. And we did this in each basin during the bucket test. So we did it on day 1 and day 3. So I'm going to show you a little video now that King County produced that gives a good example of the canine team in action.
Meet the latest innovative tools for improving water quality in King County. This is Crush, and this is Molly. Both are older animal shelter rescue dogs that have learned a pretty neat trick. They can smell the difference between human waste bacteria and other forms of bacteria, and they can smell it in water. When they smell human waste bacteria, the dogs are trained to stop and lay down or bark at the source. And they gave us a whiff of what they could do during a demonstration at Juanita Beach Park. They found the right sample every time. This can be a very helpful resource and money saver for agencies trying to determine if streams or swimming beaches have been contaminated by human waste.
Dogs have this great ability to smell and to discriminate scents that they're wanting to smell. So if something has a specific odor you want them to find, we can train them to find that specific odor and ignore the other odors.
King County and the cities of Kirkland and Seattle are using Mullen and Crush from Environmental Canine Services to inspect a couple of local waterways that have high bacteria counts. Their superior noses can help determine if contamination is due to leaking septic tanks or improper connection of sanitary sewer systems to the waterway. Other sources of contamination could be from animals, such as raccoons, geese, or beavers.
They're very good at it. Yes, absolutely. We've been all over the country using our canines to do this kind of work, and very successful at not only finding sources or getting close to the source where then managers can then come in with other tools and actually locate the source, but it's also to know where the problems are not, so we can eliminate entire branches that they don't have to investigate because the dogs say, you're fine, there's no human bacteria coming from these branches.
Water samples sniffed by Molly and Crush will also be examined by conventional and a new laboratory human source tracking method at the King County Department of Natural Resources and Parks Environmental Lab.
So after day one, we were quite impressed with the dogs. As you can see, they were pretty. They weren't very wishy-washy. It was pretty quick, rapid. They keyed on it, and they hit all our positive controls displaying on and didn't fall for any of the alternate waste controls, at least on day one. The second time we ran the bucket test, Molly, the border collie, actually keyed off of the cat do. So for those of you that are not familiar with the qPCR technology, it's not a culturing technique, so you don't have to have viable cells, and it measures the DNA in the target organism. And in this case, we were looking for Bacteroides, which is the bacteria most common in the human gut. And as I said before, this marker's been worked on by various research institutes for a couple of years now, and it's been modified and modified. And this version happens to be more specific than previous versions, which were highly sensitive but less specific, so you get a lot of background noise. But this one we're pretty happy with. So the sample is taken to the lab, it's filtered, and then that filter is frozen at -80 degrees, and then later an aliquot is processed, and then you get a data report that's quantitative in copies per mil. So I'm going to hand it over to Andrew now, and he's going to talk about the data.
So I realized that this figure is showing a lot of things, and I'm just going to try to walk you through it with the pointer here. There it is. So it's showing the E. coli concentrations in the light blue color, fecal coliform concentrations in the purple color, and the Q2 bat concentrations in the dotted line. The y-axis is on log scale, so each major breakpoint represents a change in the order of magnitude, starting with 1, 10, 100, and going up. On the x-axis, the controls are shown, the alternate waste controls in green, positive controls in brown, and the negative controls in purple. The dog hits, where they gave a positive indication, are shown in the yellow boxes and circles. So if there's one thing worth pointing out in this figure is that, as Debra mentioned, the dogs performed very well in the controls. One dog was perfect, another dog, Molly, gave a false positive on the alternate waste control. Unfortunately, they did not agree entirely with the Q2 back results. So if you look and see where the high concentrations of Q2 back are on the dotted line, it's not necessarily where the dog hits are. There's a lot of hits where we didn't have any Q2 back present. And there's also samples that had high Q2 back concentrations, but the dogs didn't pick up. So that was a little bit more discouraging. Another important question that came up was, is there a threshold of significance? So is there a magic number that if we see a certain concentration of Q2 back that warrants a follow-up action? And ultimately we came to the conclusion that there really wasn't a specific point that we reached that we had to take follow-up action on. You really had to evaluate it as just another line of evidence, and that you had to consider it in relation to other samples that were taken, the concentration of other samples taken throughout the watershed, and other factors. So these are just the results for Thornton Creek, and fecal coliform was not sampled in Thornton Creek, which is why you're not seeing it here. There were a lot of results that had high concentrations of Q2 back as well as E. coli. If you plot those on a map of Thornton Creek, a lot of them were clustered in the same area. So this map shows where all samples were taken and where all the dogs were sniffing the samples. The points represent the Q2 concentration, so the size is, the larger the size, the higher the Q2 concentration. The E. coli is indicated by color, and you can see the scale onto the right side of the figure. The dog hits are also shown in black X's and crosses. So this tributary to Thornton Creek, Little Brook, had a lot of high concentrations of Q2 and I'll go ahead and put those numbers up, upwards of 5 million, and to provide some perspective on that number, that 4.9 million is greater than a lot of the positive controls that we had, the 1, 2, and 5% diluted influent. So obviously a lot going on there. And then in the lower part of the watershed, there was also some relatively high numbers. So there was some follow-up work done this fall. 2 sets of samples were taken. On October 8th, a baseline sample, so a pre-storm sample was taken, and these are the E. coli numbers. Then about a week later, we had over ½ an inch of rain, and the same sites were resampled again. And you can see the numbers jump way up, especially in some locations, like on Little Brook Creek. There was one location, the mouth of Mock Creek, where you had a fairly high base flow concentration of E. coli, and then you did not see an increase. It was actually a slight decrease, which the people at SPU suspect is a leaky side sewer. And we're still waiting on getting some Q2 results in for this. So these are the results from Juanita Creek. The concentrations were not as high for Q2 and E. coli, but there still were some relatively high samples. Also, since it came up in the last session, it's worth pointing out that there's a pretty strong correlation between fecal coliform and E. coli that we saw in this basin. So this is same figures or same symbology as the map in Thornton Creek. And you can see where the dogs keyed in on were not where we were seeing our higher concentrations for the Q2 Follow-up work was done in these areas. So this was a residential development next to 405, and then a commercial development where the dogs were sniffing catch basins and worked back up to a frontier communications facility. And so dye testing, smoke testing, and CCTV work was done at both locations, and those sources were found. And then, of course, when we got the lab results back, we saw pretty low E. coli numbers and very little He2 present. So it's hard to say really what the dogs were keying in on there. Both of them were giving positive hits, so feel confident they were smelling something, but it's not clear what that something was, and it definitely doesn't look like it was the something that we were looking for. So these are the He2 numbers, some higher numbers in the West Fork of Juanita Creek. Also a very high number at the mouth of the Firwood tributary close to Juanita Beach Park. Again, similar to Thornton Creek, follow-up work was done in the fall where base flow samples and storm samples were taken. The base flow E. coli numbers pretty low with exception of that Firwood tributary, the mouth of the Firwood trib. Then over ½ an inch of rain, and the resample, the numbers jump up again. Similar to Juanita Creek, we see that number at the mouth of the Firwood tributary go down. The City of Kirkland's done follow-up work there, and they suspect that it's coming from some failing septic systems. So you can see that we're waiting on Q2 results here too, so that could provide some more insight. So.
All right, I'm just going to summarize pros and cons of each of these techniques that we tried, starting with the canine scent tracking team. The pros were that they were really rapid in the field. You could cover a lot of ground, and it was, if you weren't collecting bottles along with it, you could really utilize the fact that they were coming from out of town and just go for it. They were good on the controls, at least the positive controls. There was that one exception where one dog keyed in, gave a positive result on the cat do. And then, so we're thinking that maybe they might be more useful if you know that you have a really you're close to an illicit connection where the concentrations are going to be higher. Maybe not so much when you're dealing with surface water, that it's more dilute. The cons are there was variability between the dogs. So if you didn't have 2 dogs at a time, you'd want to have a lot of confidence in the one dog that you hired. Again, as Andrew had said, Kirkland did a lot of follow-up testing in those areas that the dogs were they were giving very strong positive hits, but nothing was showing up. So we're not really sure what they were keying in on. And it didn't correlate well with any of the laboratory results, not even the fecal coliform or E. coli data. And then because we don't have a locally trained dog, there was the added cost of paying for their transportation and getting them up. Pros for the human Bacteroides method was the controls all worked out perfectly. It's specific to the human gut bacteria, and you get quantitative results. So instead of just a single hit that the dog say it's present or absent, and you can see in that map that Andrew showed on Thornton Creek, you could actually see where the values were greater going in one direction or another. And then the added bonus is that you can freeze the filters and not analyze them. So you can go out and do a massive investigation, collect the bacteria data, and have it prepped for the qPCR work. The filters can be frozen, and then wait for your E. coli or fecal coliform results to come back, and then only process the ones that had really high bacteria, if you were choosing to do that. We're actually trying that technique on our swimming beach monitoring program a little bit this year, because we have a couple beaches that had continually needed to be resampled and were closed, and we don't know if it's a shallow beach, and it was hot this summer in Seattle, so we don't know if it was waterfowl, is it, dogs, is it diapers. So we're going to try that. The cons were there was some interference, and in Juanita Creek we weren't able to get results on 11 of the samples, and 4 of those being the alternate waste samples. We don't know why. The lab tried to explain it to us, but it's not very clear. And the DNA can last in the environment up to a week, depending on the conditions, and that may be a con, depending on if you're looking for really fresh, culturable bacteria. And at best, it's a 2-week turnaround time, and that's on the optimistic side right now. So there were a lot of people that helped on this project. It was a big team effort, and I'm not going to name all these people, but you know who you are, and some of them are in this room. So this is I think we're holding off on questions until Jeannie's done, but this shows the example of just how many bottles and bottle kits were processed. I just have to give a thumbs up to our lab for preparing all this. They were pretty incredible.
Thank you. My name is Jeanne Dorn again. I work for King County Stormwater Services, and I'm here today to talk about what are we doing for fecal coliform TMDL. And our fecal coliform total maximum daily load allocation program that we're working on right now, we've been tasked with looking at our creeks flowing into the Puyallup White River. And the 2 creeks that are in unincorporated King County that discharge the White River are Boise Creek and Jovita Creek. So what we've got to do is we've got to look for sources of bacteria, high levels of bacteria, and work to eliminate them. And it's a multi-pronged approach, 3 general areas that I've separated into different components. Research, look at what's available. Technical methods, of course, which we've been just talking about, and very importantly, building and maintaining good relationships. So jumping to what the Boise Creek Basin looks like and where it is, it's located here's a map of King County, and the Boise Creek Basin is down in very southern part of King County, just near Pierce County. And it's a basin that is typified largely, at least in our area of concern, agricultural rural basin, very flat, very high groundwater year-round, lot of roadside ditches, and with some piped conveyance. The people approach, which I mentioned is very important, is we've been working very hard to build new relationships, leverage existing relationships to get things done, form partnerships, work to gain trust, so that we're all working toward common goals. And I'm not going to read all these out, but these are all the different groups that have been involved in this effort of bacteria screening and eliminating sources. Of course, I'm in King County Stormwater Services, so I'm starting with that group first, just to let you know what we've been doing. As far as the people approach, what we've been very busy doing is attending and participating in public events in the Boise Creek Basin. As many times as possible, we talk about our mandate to find high sources of bacteria and to eliminate them. And very importantly, working to gain the public trust and other agency trust. If you say you're going to get back to a person, get back to them. The people approach is the most important. A group we've been working successfully with down there is King Conservation District. King Conservation District is the old soil conservation service, which was formed in this country 75 years ago during the Dust Bowl. Here in our area, it's called King Conservation District, and they've been here in our area 65 years. So these people, King Conservation District, have been working with farmers and others in Boise Creek area, working with them on a voluntary basis, real positive. They're the ones who issue farm management plans, or they're just called farm plans, which farmers voluntarily implement. So they've got a real positive relationship with the people in the basin. And they also recently they've instituted what's called the Stream Steward Program, which is an effort to get citizens involved in monitoring and taking water quality samples in Boise Creek. And King County has supported that program by providing some training and also providing some equipment. And King Conservation District, as I've got here, can help landowners directly improve their best management practices. The goal is keeping water runoff clean. And part of the people approach component is just stormwater services person there, whoever that is, at a stream steward training, another King Conservation District event we attended, and a King Conservation District fair we attended. This is Ray Tim of our science group, and there's a drenched stormwater services person, Jeanne Dorn there, and literally getting drenched, but that's what I'll do for King County. And the people approach, again, Stormwater Services has been working very closely with our own King County Science and Technical Support Section, helping us figure out what are the new techniques and what do they mean, and Ray has been very helpful in performing actual field recon and sampling in the basin. We are so blessed to have a great environmental lab at King County. They've helped us learn more about the microbial science, which we've been talking about today, the qPCR techniques, and they've helped with me scheduling sampling, and just thought I'd throw this in. Isn't this an awful place to work, right on a beautiful canal near Seattle Pacific University? That's our lab. It's a fantastic lab. Wonderful people work there, do great work. Another agency we've been working real hard as Stormwater Services, we've reached out to another King County group, a Rural and Regional Services group, which also work to have positive relationships with farmers. So they help with getting cost share projects implemented on farms similar to what the KCD does. They work with the King Conservation District. And another group we've built strong relationships with in the last couple of years is our own Public Health Seattle King County. I'll get it right. And they're instrumental in our work because where could some high levels of bacteria be coming from? Septic systems. Well, this is the agency that approves and inspects septic systems, so very important to have good open lines of communication with them. And this is over on the left here is what, of course, we don't want to see, and over on the right is what we do want to see. And just a little side note, I love septic systems because septic systems put the water back into the groundwater table. When it percolates through the soil as it's supposed to, the soil and the good bacteria clean the water. We want it to go back into the groundwater and return to our streams as base flow, as clean water. If everybody in the world was on sanitary sewers going through a wastewater treatment plant, that would not be good for a worldwide ecology. So we love properly functioning septic systems. Washington State Department of Agriculture is another part of our people approach. They're in the basin and they deal with dairies. So I've gone to them with some concerns and they've listened. It sounds like I've been on the phone a lot, and I have, I guess, and on the email a lot. Washington Conservation Corps is another group that I've been working with. And I haven't been working with them in Boise Creek directly, but these are the people in King County with Washington Conservation Corps workers. They do things like stream buffer restoration along creeks, and this picture is of the nearby basin, Newaukum Creek, where there's been a lot of restoration efforts going on. And it's possible, if there's money and time and all that available, maybe they can do some work in Boise. So that's a good group to reach out to. So now moving into the technical approach. The technical approach, what did we do when we started working on this? I first thought, well, let's use existing data. So let's do a sort of a desktop GIS analysis. We just looked at air photos and tried to identify possible areas that would be discharging bacteria, like manure lagoons, places where animals are accessing ditches or creeks. And we came up with a nice preliminary map, which I've kind of put to the side because I've dived into other things, but I might revisit it and try to make sense of it. The other thing that I've been doing, along with my fellow stormwater services people, working with sciences. Ray Tim specifically is a scientist who has been working in Boise Creek, taking a lot of water samples over the last few years. And the yellow and green dots here are Ray's sampling stations, and he's been accumulating a lot of analytical data. So trying to look at that, make sense of it, and build off of it, because I'm not in the creek unless I slip and fall in. I'm in the stormwater conveyance system that feeds the creek. So we're kind of jurisdictionally, that's how Ray's work and mine is kind of artificially separated, but there it is. So the technical approach that we've been doing, just continue, is there's no substitute for actually getting out and driving and walking all over the basin in all kinds of weather, not just dry weather, in wet weather many times, to get an idea of what's going on, and I'll get into it a little later, to find illicit discharges. We do use some technical apparatus. I grab samples for the lab, and I also have a nice hydrolab. I like to try to use the ammonium ion probe to find high levels of urine. Don't know if it's really worked yet, but we're going to keep using it and gathering data. And this picture right here is one of our interns, science group's intern, Ture and I, gathering a $2 Coliscan Easy Gel sample. $2. Anybody can run these and incubate them overnight. $2 is pretty affordable. So so far I've actually got 44. Right here it says I've got 42 municipal separate storm sewer system sample locations, our stormwater conveyance system. I've actually increased it to 44. So I have 44 locations I've been going out looking at, screening, sampling. And let me dive into the weeds just a little bit. Recently, in June of 2014, we were out there doing some catch basin lid pulling, and we saw gray milky discharge coming from this private clay tile here. And you can kind of see the milky, yucky water collecting in the sump there. And luckily I had a whole bunch of the $2 Coliscan test kits with me, so I just went ahead and grabbed a sample. Went back to our lab, not our lab, just our offices. We've got a little tiny lab that we call a lab, but I didn't have to go to our lab. The upshot is that the next morning I saw that we had a ton of E. coli growing. And the question was, okay, we got a lot of E. coli, what's the source? The question, I had that question because it was a cattle ranch with a residential system discharging to our drain tiles. So what is it? I needed to know pretty quickly. So we went back and sampled, and I sent 4 samples, or I sent a sample for 4 lab tests to the lab in Florida because I needed very quick results. Sent it for analysis for human Bacteroides and cow Bacteroides, Both of those are qPCR tests, and the results were no cow Bacteroides, so it wasn't a manure pile leaching in. It was definitely a failing septic. The health department got involved. They went out, performed dye testing, and luckily the property owners were very cooperative, and by September they had a new septic system installed and functioning. And this was about 80 feet from Boise Creek and was discharging directly into Boise Creek. So that's one success story, and we're going to continue working on that. And Juanita Creek is our other creek. It's a very different basin. It's residential, still on septic, so we will be doing the same kind of works. I know I have to go quickly. Just wanted to say that in Jovita, we wanted to know the exact geography discharging to our MS4, and so we walked out in the basin, all around the basin, and drove, and realized that outside the topographic boundary of the basin, there's actually a new residential development that has a stormwater facility that actually is piped into Jovita Creek. So if we hadn't have done that work, we would have missed, what's the actual topography of our MS4. And over on the east, we did a more accurate job, accurate delineation of the MS4. And we'll just keep looking. And thank you very much. Any questions?
Well, actually, I'd like to jump in. I have the good fortune to be Jeanne Dorn's supervisor. We just got notice that she's found a 4th failing septic system in the Boise Creek Jovita area. And in addition, we have recently been awarded the 2014 Partnership Award from the King Conservation District as the best governmental partner that the Conservation District is working with. So that's a real